the positive feedback we saw in the last slides, in the previous slides. So that positive feedback depends on the interleukin-2 receptor activation. What happens? The downstream pathway with the interleukin-2 receptor is another protein pathway, protein mediators, okay, proteins and further proteins, you know. The ultimate effect is proliferation, induction of cell cycle, okay. And the notorious or the famous target in this pathway is mTOR, M for mammalian target of rapamycin, mTOR. Okay, so sirolimus, the previous name of sirolimus was rapamycin. Okay, so mTOR is mammalian target of rapamycin. It was identified initially in the lower species. Then it was found out to also have a a counterpart in the mammalian cells, and that counterpart is now termed as mTOR, okay, in mammalian cells, including the humans. So mTOR is the mammalian target of rapamycin, okay, and rapamycin is also called sirolimus, okay. That's how you link mTOR with sirolimus, okay. So once sirolimus binds to it, there is no cellular proliferation, okay, or differentiation, okay. And once the differentiation is in place, for example, the cellular division, active division, proliferation is taking place, it becomes an actively dividing cell. And you know that the nucleotide synthesis inhibitors, like the pseudonucleotides, like azathioprine, which is actually a prodrug, azathioprine and mycophenolate, okay? So both of them, they effectively block the purine nucleotide synthesis, especially the guanosine nucleotide synthesis. Okay, so once the nucleotide synthesis is blocked, the cellular replication stops. Okay, and the same aspects were also discussed last lecture in terms of the cancer chemotherapy. So similarly, the active cell division stops. Okay. Now, here is an important thing to remember. The pyrimidine analogs, like 5-fluorouracil, okay, is important as an anti-cancer agent. The purine analogs are more important as an immunosuppressive rather than a generalized anti-cancer, a broad spectrum anti-cancer. Whenever we talk about the hematological cancers, we see again the role of the purine analog. Now, what has to do this purines with the blood cells or immune cells?
Very important point.
Salvage pathway and DNA synthesis. If you remember, salvage pathway and de novo DNA synthesis, purine synthesis, there are two complementary pathways present in all of our cells. Okay, but the immune cells are particularly deficient in salvage pathway. So they rely heavily on de novo synthesis of purines. So if you inhibit the purine synthesis effectively, the first cell line to suffer is immune cells. Okay?
They can be also used in the immunosuppressants and in...
So that's why mercaptopurine was effective in leukemias. Okay? And azathioprine is effective in as an immunosuppressant. Right? Mercaptopurine is also as an immunosuppressant in low doses. Yes, true. Clear so far? So far together?
No, only de novo and the immune cells.
Sorry?
Only de novo synthesis and the immune cells.
The salvage pathway is not that much important. I mean, it's not very prominent in the immune cells. The they heavily rely on de novo synthesis. So if you block with azathioprine or mycophenolate the de novo synthesis, there is no way to recover. Okay? Did I move on? Clear? Okay, so what happens if mTOR dependent signaling is blocked with the sirolimus? Now, mTOR is found in a number of immune cells. Okay, here we see the macrophage. If it is macrophage, the TNF and interleukin 6 and interleukin 12, which are important in recruiting the immune response, etc., they will be blocked. So the macrophage-related reactions would also be blocked if you block it with the rapamycin or sirolimus. So bacterial immunity would be compromised. Viral immunity would be compromised because there won't be any memory T cell formation if you block mTOR. And for the regulatory T cell, if you block it with the rapamycin or sirolimus, there won't be any induction of regulatory T cells. Clear? Okay, good. So let's now move on to the individual drug account. Cyclosporine is a lipophilic polypeptide. When you talk about polypeptide, that means it's a protein.
It's lipophilic.
Sorry? It's lipophilic. It's lipophilic, can cross various tissues and widely distributed in the system. Can be given oral or IV. Okay. Hepatic metabolism and clearance. Immunomodulator. Okay. There is immunosuppression as well as sometimes immunomodulation in terms of the graft rejection. Inhibits antigen-triggered signal transduction in the T cells, blunting the expression of lymphokines. And we know now because of its ability to bind cyclophilin, calcineurin pathway. The most important toxicity is nephrotoxicity. Okay? And the most peculiar toxicity or adverse effect is hypertension and hyperkalemia. Okay, not precisely understood why, but there is some element to be attributed to nephrotoxicity, okay, which leads sometimes to hyperkalemia and hypertension. But nephrotoxicity alone doesn't explain the reason of hypertension. Okay? So hypertension, hyperkalemia, they are the most peculiar adverse effects of cyclosporine, and nephrotoxicity is the most important adverse effect. Okay, most feared adverse effect. It is teratogenic and it inhibits the metabolism of glucocorticoids, so if given with the glucocorticoids, would be synergistic. Then you have more and more immunosuppression if used together. Since it is nephrotoxic, it can add to the nephrotoxicity of other nephrotoxic drugs, like NSAIDs. Okay? And it competes for its plasma protein binding with methotrexate, so it can displace methotrexate. Okay? So there would be a higher free methotrexate in plasma. Need to reduce the dose of methotrexate. Most of the adverse effects of cyclosporine are dose dependent. So in the transplantation centers, graft transplantation center, grafting and transplantation facilities, they should have a way to monitor the plasma levels of cyclosporine, because most of the times they use this drug as a graft for the graft survival, as an immunosuppressant. Okay, and most of the side effects are dose dependent. Or you can say plasma level dependent. Okay, so you need to monitor plasma level. The second drug is tacrolimus. Tacrolimus also works through the the immunophilin, the calcineurin pathway, but it is better in terms of efficacy and better in terms of the side effect profile. So fewer or lesser in toxicity, better in efficacy. So it's advantageous. It's a macrolide, and I understand you remember what macrolides are. Like, yes, so macrolide just like erythromycin and azithromycin, so they are macrolides, okay? So this is also a macrolide. It could be given oral or IV. Should not be given with food. Food retards its oral bioavailability, okay? If given orally, bioavailability is reduced. And this should be given empty stomach or substantial amount of time later if the food has already been taken. Okay, maybe a couple of hours later. Hepatic metabolism and excretion. You know the mode of action. You know this aspect. You know this also. So we will be emphasizing only the differentiating points. It's easy to remember. Sirolimus or rapamycin is the related drug. It's another macrolide, but it doesn't work with the calcineurin. It works with the mTOR. Could also be given the same way. Same way. This is different. This is also similar. In addition, it can produce dyslipidemia. So dyslipidemia is more marked than the catecholates. Okay, adds to nephrotoxicity, similar to others, and it can increase cyclosporin levels. Okay, can inhibit metabolism of cyclosporin. So, if you're using it with sirolimus, reduce the dose of cyclosporin.
So it reduces the effectiveness
It increases levels. That means free cyclosporin will be more. So reduce the dose of cyclosporin. Okay. Now this is the other group of drugs now starting, the ones which tamper with the purine metabolism, and as this is already you know, the de novo synthesis salvage are the ones through which we get the active purines, and the breakdown pathway which leads to the generation of uric acid. Okay, this is the one which is there to degrade the purines. Yeah, this is all.
That's the de novo.
You know. Okay.
Mercato periods.
So now we are launching on the purine metabolism and the sites of drug action. Okay. Azathioprine, as you already know, is the analog, is the prodrug which generates 6-heptopurine. It's a purine. Okay. It resembles the normal purine. Hence, it uses the normal body enzyme machinery to synthesize the precursor purine nucleotide. What is the precursor purine nucleotide normally? Inosine monophosphate. Remember, inosine monophosphate is the common purine which further is modified in either to adenosine monophosphate or guanosine monophosphate. IMP changes into AMP or GMP. If you remember biochemistry. So since it's a sulfhydryl containing or thio group containing molecule, so it synthesizes thio IMP. Okay, which is not natural IMP. Okay, so TIMP or TIM is thioinosine monophosphate. Here it is, and this thioinosine monophosphate would form 6-thioguanosine nucleotide. Okay, 6-thioguanine nucleotide. Right? Clear? Oh, you are more interested in attendance. Okay.
This is the first time.
Please pay attention. Okay. They have to discuss some questions as well. If you want to spare some time, you have to pay attention so that I can finish it. Okay?